Hello, everyone. My name is Matthew Thompson. I am an assistant professor in the Department of Chemistry and Biochemistry at the University of Alabama. I'm going to give you a brief introduction to my research in my research lab today. If you have any questions about anything I talk about, or if you just want to know more, shoot me an email, mthompson10@ua.edu. You can also just go to our website, thompsonlab@ua.edu, look us up, and if you can't find what you need there, shoot me an email. So there are actually two projects that we work on in my lab. Unfortunately, for this brief introduction, I've only got time to talk about one. So I am going to talk to you today about the fosfomycin resistance project in our lab, or antibiotic resistance. We also work on electron transfer in DNA processing enzymes, and you can go to the Thompson Lab at ua.edu and get a little bit of information on that. So you may be aware that antibiotic resistance is one of the most pressing issues in modern medicine. This timeline here across the top, this shows when antibiotics, it points to the year when antibiotics were introduced or deployed into the clinic. And then on the bottom, it points to the year where resistance was observed. And so you can pick out some familiar ones like penicillin, introduced in the 1940s, and then in 1945, resistance was observed. Maybe you've had ampicillin or methicillin. Here's methicillin. You've heard of methicillin-resistant Staph aureus, maybe. It was introduced in the 60s and or in 1960, and then about 1962 or 1963, resistance was observed. And really, what you see is it's almost a mirror with respect to the antibiotics listed on the top and in resistance on the bottom. And so there's currently resistance to every single antibiotic that we have deployed. Now, one of the things that you see, or I'll point out, is this kind of gap of about 30 years where no new antibiotics were introduced. And so really in the 1960s and 1970s, medically, we were really enjoying life and having antibiotics and thought we had the problem solved. But now we know that we didn't, and we didn't make any new antibiotics, and resistance just kept creeping up. And so we've gone back now. There are some new things starting to come out, and maybe even a little bit later here. But we've got to figure out what to do about this antibiotic resistance problem. One of the strategies that we use that's common is combination therapy. So many of you, if you're not allergic to it, you've probably had something called amoxicillin. When I was a kid, I had it. It was like a pink liquid that my mom kept in the refrigerator. Now it comes also as a tablet, but amoxicillin now is not administered by itself. It's administered with a second compound called clavulanic acid. So amoxicillin is the antibiotic, all right? And amoxicillin targets this protein called PBP, or penicillin binding protein. Penicillin binding protein, this enzyme here, is involved in peptidoglycan biosynthesis. And so a bacterial cell wall is made of peptidoglycan. If the bacteria can't make a cell wall, they can't survive, right? So this antibiotic shuts down bacterial cell wall biosynthesis. But be it as it may, evolution wins out, and the bacteria evolved another enzyme called beta-lactamase. So now, when bacteria are challenged with something like augmentin, they will overexpress an enzyme that comes and binds to augmentin and then clips what we call the beta-lactam ring in here, and it destroys the antibiotic. So what have we done? We have discovered a second compound that inhibits beta-lactamase. So now we give both of these compounds together, and the first compound, amoxicillin, is the antibiotic, and the second compound is a small molecule inhibitor of the enzyme that destroys the amoxicillin. So if this enzyme doesn't work, then the amoxicillin can do its job. And this is called Augmentin. Some of you may recognize it. I think the last time as an adult that I was prescribed amoxicillin, I actually got Augmentin. So this is combination therapy. And this is an example of what we're trying to do in my lab. We don't work with amoxicillin. We work with a small molecule called fosfomycin. All right, so this is an antibiotic, and I really liked this antibiotic because just some simple things about it. It has no toxicity in humans, no toxicity whatsoever in humans. So when you take this stuff, it's eliminated from the kidneys in its active form. It's not metabolized at all. This stuff goes right through your system. So we prescribe it as like a single 3 or 4 gram dose. And if it's not metabolized, then it builds up in your urine before you excrete it. And so it gets to really high concentrations in your urine. And that's why we typically prescribe it for urinary tract infections or UTI. It's marketed in the United States under the trade name Monorol. Some of you may have had it. The problem with fosfomycin is that the enzymes have evolved, the bacteria have evolved an enzyme that destroys fosfomycin. So when we challenge bacteria, and this enzyme actually is from MRSA, or methicillin-resistant Staph aureus, when we challenge something like MRSA with fosfomycin, MRSA overexpresses this protein or enzyme that binds to the antibiotic. Here's fosfomycin. Binds to the antibiotic. And then it takes a thiol, an SH, removes the H, and uses the sulfur as a nucleophile, and it attaches a thiol. And this is L-cysteine. This is bacillothiol, BSH. You'll notice that bacillothiol it has a cysteine on it, right? Cysteine has a thiol. So the enzyme takes a thiol and catalyzes nucleophilic addition of the thiol to fosfomycin, and this results in a compound that has no bactericidal properties. All right, so here is the equivalent enzyme to fosfomycin as beta-lactamase was to beta-lactam type antibiotics. Now, one of the things that we've done is we can do a simple disc diffusion assay. And so here you take a Petri dish. Just look at this top one. You take a Petri dish and you grow something like MRSA up in it. This is methicillin-resistant Staph aureus. You grow MRSA up. And then you take these little white pieces of paper and you've soaked these little white pieces of paper in your antibiotic and you drop them onto the Petri dish. And when you drop them onto the Petri dish, then the small molecule, in this case the antibiotic, starts to diffuse out into the media. And as it diffuses into the media, it starts to kill the bacteria. And you get this sort of clear area that starts to form. We call that the zone of clearing. You're basically trying to see how well your antibiotic kills the bacteria as it diffuses into the media at different amounts. So here's 10, here's 25, 50, and 100 micrograms of the antibiotic on each one of these white pieces of paper. And so this is what we call wild type MRSA, meaning we haven't done anything to it. It's the native strain. When you put these little discs with the antibiotic onto the Petri dish, it doesn't really kill much. These zones of clearing are quite small. By comparison, let's look at, if you just go with the second one down, right? Here is where we have taken the wild type MRSA and we've knocked out the DNA that encodes for this protein, right? So now you're growing up methicillin-resistant Staph aureus, but it doesn't have this enzyme. It can't produce this enzyme because we've knocked out the DNA for it. And now when you take the same white discs and you drop them on there, you can see that it practically clears the whole Petri dish, right? Whether it's at 10, 25, 50, or 100. So even at 10 micrograms, the zone of killing has gotten much larger. Now this second experiment, we knocked out the gene that in one of the enzymes that produces the second molecule. Remember the enzyme takes phosphomycin and adds this small molecule to it. So whether we knock out the enzyme or we knock out the small molecule or production of the small molecule, you can see that when we challenge MRSA with phosphomycin now, we basically clear the whole dish. So this validates this protein or enzyme called FosB, validates it as a drug target. So we can see that if we inhibit this protein and shut down this protein's function, maybe with a second small molecule, we would be able to use phosphomycin to fight MRSA. And that's exactly what we're trying to do. So we take the protein structures in our lab, we obtain the protein structures in our lab, and we use some really neat computer programs that allow us to take small molecule libraries, and these computers take our protein structure and millions of small molecules, and the computer program tries to fit the small molecules into the active site of the protein, and it scores them for us. So we can screen millions of compounds very, very quickly. It's called high throughput screening. It's basically computer-based high throughput screening. We identify compounds. Now, we don't know if they're going to actually inhibit because of the computer program doing it, right? But we can do a couple of checks to make sure that it's going to be potential drug candidate. And then we just buy them, and then we test them to see if they actually inhibit our protein. We can work out the full inhibition kinetics, and then we're a structural biochemistry lab, so then we want to see, we want to get a protein structure with those small molecules bound, so we can see if the computer program actually got the orientation of it in the active site of the protein right. And once we have a crystal structure of it, then we can maybe look at how the small molecule is bound down in the active site, and then maybe we can do some chemistry to modify the small molecule and improve protein ligand interactions, and maybe through some rational design and synthesis, we can even make it a better inhibitor and make it bind tighter to our protein, and then we can go back and do those inhibition kinetics again, and get all the kinetics worked out, and then maybe we can get a crystal structure, and then maybe we can improve, modify the compound a little bit more, and you go around this cycle a few times until you really have obtained a potent inhibitor of your protein, and then maybe you go back into cell-based assays and maybe even into animal models. This is animal models. And so that's what we want to do. We want to design not necessarily new antibiotics, but find inhibitors of these FosB and FOS. There are other classifications of these enzymes, and recycle phosphomycin and maybe turn it into a usable antibiotic. So the things we do in the lab then obviously are high throughput screening. We do a lot of enzyme kinetic assays, and then one of the big things for us is protein crystallography, growing crystals of proteins and sending them to the synchrotron and getting high resolution structures. And then we have collaborators that help us with the organic synthesis. If it's not too hard, we can maybe do it in our own lab. So there's a lot of different things that students would get to learn how to do in a project like this. So once again, if you have any questions, shoot me an email. This was only part of our research. If you want to see the rest of it, thompsonlab.ua.edu. And I look forward to getting to talk to some of you on February 13th.